Hello, in this video, let us see immunity in a nutshell using a case. The case follows like this. A mutated viral protein has entered the body of an individual who is showing mild respiratory symptoms. The immune response against the intracellular foreign protein is best described by which of the following simplified sequences? The options are A. Proteasomal digestion of the protein, that viral protein in the host cell. Then the immune synapse with the CD8 T cells along with the MHC class 1 peptides. Then the activation of cytotoxic cells, ultimately host cell destruction. This is option 1. Option 2, B. Antigen recognition by the macrophages. Immune synapse with CD8 cells along with MHC class 2 peptides, activation of cytotoxic cells, immunoglobulin secretion. C. Immune synapse with the memory B cells along with MHC class 2 peptides, proteasomal digestion of the protein, this is viral protein, activation plasma cells, inactivation of foreign protein. D. Proteasomal digestion of viral protein, immune synapse with the T cells along with the MHC class 1 peptides, activation of antibodies, then antigen antibody reaction. These are the options. Let us see which one is the correct. To understand this, we have to go to the immune processes. Let us see the immunological response to a bacterium and then to a virus. We have to follow the numbers, labeled numbers. So there is a bacterium which has entered the body. Number 1, it is phagocytized by the antigen presenting cell. This is going to present antigen, that is a bacterial antigen. It is phagocytized, this is the macrophages. It has MHC class 2 proteins on the surface. So along with the antigen, that is digested bacterial product protein, the APC presents the bacterium number 3 to the helper T cells. There is a immunological synapse with the helper T cells, MSC2, and the bacterial protein is presented to the helper T cells. The antigen presenting cell secretes interleukin 1, which makes the helper cells proliferate, activate. So there's a immunological synapse here. This is helper T cell, and helper T cell secretes interleukin 2, which has an influence on itself. This is called as the CD4 cell. The CD4 cell now activates the cytotoxic T cells. These are all different types of T cells. Helper T cells we have seen, cytotoxic T cells, which is also called as a CD8 cell. Number 7 is the viral protein. Here comes the viral protein, which is similar to our case. The virus infected cell. This viruses always infect the cells, living cells. They go into the RNA or DNA, and then this virus infected cells is presented to the cytotoxic cells along with the MHC class 1 protein on the surface. So there's an immunological synapse, and ultimately there will be a destruction. So this is the cellular immunity, what we have seen. The bacterium is subjected to the cytotoxic cell for destruction. The virus is subjected to the cytotoxic cell. This is the cell mediated or cellular immunity. Now let's go to the other type of immunity. There is a humoral immunity. Helper T cells, we'll go back to the helper T cells number 5, and we'll go on to the 8. So helper T cells secretes interleukin 4 and 5, activates the B cells, B lymphocytes. Now come to the B lymphocytes. 9. The B lymphocytes gets transformed into the plasma cell by blastoid transformation. Number 9. And the the plasma cell secretes immunoglobulin, various classes of immunoglobulin. 10. And immunoglobulins react with antigens which has entered by antigen antibody reactions. And the memory cells are also formed at the same time. 11. One more stage, bacterium also can be subjected to the B lymphocyte directly. The B lymphocytes can also be antigen presenting cells. This is the immunity in a nutshell, from 1 to 12. It's a summary of acquired immunity, as a specific acquired immunity. You can follow the sequence in the next slides by clicking on the number links. Let's go to the same thing once again. Antigen presentation by the macrophages. Antigen presenting cells, mainly the macrophages, partially digest antigen. Here the bacterium present part of the antigen, the protein, along with the MSC2 peptides on the cell surface. We can go back to these steps again. 1, 2, 3. 1, 2, 3. Next is helper T cell activated, produces interleukin 2. Interleukin 2 acts in a feedback manner, autocrine manner, and stimulates the multiplication of the helper T cells. Stage 5. This is the stage 5. Autocrine secretion of interleukin 2. The proliferation of helper T cells. Next is the clone of helper T cell is formed, a large number of. Next, stimulation of cell-mediated immunity. The activated CD4 cells in turn activate the cytotoxic CD8 cells and thereby triggering the cell-mediated immune response. This is the one. Follow step 6. Interleukin 2 by the helper T cells not only has an autocrine effect but also activates cytotoxic T cells. These cytotoxic T cells have various cytotoxic or cell damaging effects, including the damaging the bacteria. Now, CD8 cells that can also be activated by forming immune synapse with the MHC1 cells for virus infected cells. For the virus infected cells, this is the step 7. So, virus infected cell will be destroyed. This is also the cellular immunity. So then virus infected cell undergo lysis by insertion of the perforins in the cell membrane, and the virus cell along with the virus undergoes destruction. So cellular immunity is mainly a defense against viruses, fungi, tumors, and also involved in the graft rejection. Now humoral immunity, activated CD4 cells helper cells activate B lymphocytes by secreting interleukin 4 and interleukin 5. This is 8. This one. The B cells undergo blastoid transformation, multiple, and form clones of plasma cells. Plasma cell, a large number of plasma cells are formed, which secretes immunoglobulin. Plasma cells produce antibodies. Step 10. This is aimed primarily at bacterial infection, cellular immunity. We also see in the formation of memory cells, immunological memory. This is the one. 11. Follow step 11. Following antigenic exposure, a considerable number of activated T and B lymphocytes persist as memory cells, both T and B. So cellular immunity, humoral immunity has a immunological memory. So later exposure to the same antigen, bacterial or viral, causes these memory cells to get activated to effect our T or B cells very rapidly. So either humoral immunity or cellular immunity develops very rapidly and with a amplified manner. This is immunological memory, and this is the basis of the vaccination. There is the active immunization, vaccination. Giving vaccines, we have heard of COVID vaccination. So immunization develops actively by antigenic exposure through the form of a vaccine. The body has already memorized. The first entry, later entry will cause an amplified and rapid response. As we have seen, antigen presentation can also be done by the B lymphocyte. B lymphocyte themselves can present antigens. They recognize antigen and present them to the T cells, helper T cells. Helper T cells have to be contacted by these APCs for full activation of humoral immunity. As such, B lymphocytes, when they contact antigen, they cannot multiply unless helper T cells are contacted. This is the 12, step 12. So we will find the answer for the first question we have. So the case mutated viral protein. That means a new variant has entered. That means it has not entered previously. Variant is new. So it should be the first exposure. So the immunological response against this now can be, this is the answer. Proteasomal digestion of the protein in the host cell. Immune synapse with the CD8 T cells along with MHC class 1 protein. Activation of cytotoxic cells. Host cell destruction. These are not in sequence or they are wrong. Antigenic recognition by macrophages. It is a viral protein. Mainly it is intracellular organisms. So unlikely that macrophages recognize. Immune synapse with CD8 cell along with MHC class 2 peptides. Activation of cytotoxic cell. Immunoglobulin secretion is wrong because cytotoxic cells as such, they do not secrete immunoglobulin. So immune synapse with the memory B cells. Memory B cells do not synapse with this. MHC class 2 that proteasomal digestion protein activation, plasma cell inactivation. This is some vague, it's not in order. Proteasomal digestion viral protein, immune synapse with T cells with the MHC class 1 protein, activation of antibodies. Antibodies cannot be produced by these T cells, they have to be from B cells. So this also will be invalid. Partially, these are all partially correct. The correct answer is A. Proteasomal digestion. A protein in the host cell. That's why it has entered the host cell, immunologic synapse with the CD8 cell, along with MHC class 1 peptides, activation of cytotoxic cells, host cell destruction. That is this one. This is the last, when the virus contacts with a variation or variant form. This will be the immunological response.